Welcome to the fourth video in my series on antimicrobial drugs. In this video, we'll be talking about the antifungals and antivirals. Your main objectives for this video are to pay attention to the drugs that I talk about and try to make notes about how they work. For example, you should be able to describe how antifungals like polyenes, azoles, echinocandins, and nucleic acid inhibitors work. You'll need to know their targets and what their effects are. You should also be able to describe two types of antiviral drugs and give an example of each. Even though I didn't put it here, it wouldn't hurt to know what kinds of viruses usually are treated with antiviral antibiotics. You'll also probably want to pay attention to the interferons. So far in our discussion of antimicrobials and antibiotics, we've spent the majority of our time talking about antibacterials. Bacteria are prokaryotes, and as we learned in Chapter 4, there are many characteristics that distinguish those cells from eukaryotic cells like our own. For example, they have peptidoglycan and 70S ribosomes. They have different enzymes related to nucleic acid synthesis that are not shared with eukaryotes. This means that there are more magic bullets available to treat infections caused by those types of cells. But what about fungal infections? Fungi are eukaryotes, just like humans are. As a result, there are many characteristics that we share with fungi, making treatment a bit trickier. Why is treatment trickier? Well, there are fewer magic bullets. This table shows just a smattering of the similarities that we have with fungi. That said, there are two popular targets for antifungal drugs. These targets are ideal because they're present in fungi, but not in human and most other eukaryotes. These drug targets are the cell wall, which is made up of chitin, and ergosterol, a fungal sterol. Ergosterol is a steroid alcohol that gives stability to fungal plasma membranes, much in the way that cholesterol helps with stability of our plasma membranes. Polyenes are a group of antifungal molecules that bind ergosterol in the fungal plasma membrane and cause the formation of cylindrical pores that allow cations and eventually large molecules to actually leak out of fungal cells. This leads to fungal death. Amphotericin is one of the oldest and most famous antifungal drugs. Why? Well, primarily because of the severe side effects that it can cause, such as kidney failure, liver failure, and heart attack. Milder side effects include fever, chills, difficulty breathing, and blood pressure changes. For this reason, amphotericin is also known as amphoterrible, and it's primarily used for life-threatening systemic mycoses. Amphotericin is only available in medical facilities like hospitals because it's administered by UV for systemic infections, or it can be given orally for thrush. Thrush is an oral candidiasis infection. Nystatin is another polyene that's preferred to amphotericin, but it's still extremely toxic and is only used topically in ointments or orally to treat candida yeast infections. There's no injectable form of nystatin because of how toxic it is. Now you might be wondering why these drugs are so toxic. Well, just as they form cylindrical channels in fungal cell membranes, they can form them in our membranes as well, particularly in the kidneys. Azoles are alternatives to the polyenes. These drugs were only developed in the 1970s, and they inhibit ergosterol synthesis. As you can see from this image, well, kind of. Ergosterol synthesis is the result of about 6 different chemical reactions. Azoles bind an enzyme in the pathway that catalyzes the final conversion to ergosterol, so a toxic sterol is synthesized instead. This, when it inserts in the plasma membrane, disrupts the membranes of fungal cells and inhibits growth. So you can see they're trying to show that here. Some examples of azoles include clotrimazole, or fluconazole, or miconazole, or ketoconazole. These can be purchased in some well-known antifungal treatments available at your local drugstore. Azoles are used primarily topically to treat superficial mycoses like athlete's foot, ringworm, and some candidiasis as well. That said, certain azoles can also be taken orally to treat systemic infections. Alliamines are an alternative for azole-resistant infections. Developed between 2001 and 2006, echinocandins inhibit an enzyme involved in beta-glucan synthesis. Beta-glucans are complex carbohydrates that, along with chitin, form the backbone of the fungal cell wall. The addition of other polysaccharides further stabilizes the wall and gives the fungal cell extra support. In the presence of echinocandins, however, those beta-glucans are not cross-linked to the chitin cell wall. This makes the cell wall become very weak and fungal cells become susceptible to osmotic lysis. Sound familiar? Yes, that's why the echinocandins are called the penicillin of antifungals. Typically, this type of drug is used to treat candidiasis and aspergillosis. You might have seen griseofulvin in someone's medicine cabinet. It's a drug that's used for treating superficial dermatophyte infections. Most commonly, you see it as a treatment for nail infections. The way that this drug works is it inhibits microtubule formation in fungal cells. Do you remember what microtubules are? Remember, these are the protein rods that form the cytoskeleton in eukaryotic cells. But where it's important for this discussion is that these protein rods also form the hangers that eukaryotic chromosomes are hung on during mitosis and meiosis. So if we disrupt microtubules, cells lose their ability to separate their chromosomes during mitosis and meiosis. That inhibits growth. Tolnaftate is another antifungal drug. We don't really know exactly how it works. We think it may inhibit ergosterol synthesis by activating a required enzyme. You'll find this drug in a product called Tinactin, and I remember some very old commercials where they used to say that Tinactin was tough-actin Tinactin. And that commercial always stuck with me, even though that was like a billion years ago. Nucleic acid synthesis is the final drug target for antifungal drugs that we'll be discussing. Flucytosine is called a prodrug. That means it's administered in an inactive form, and it's not until the drug actually enters the cytoplasm of a fungus or a bacterium that it becomes active. It gets converted into a nucleotide analog called 5-fluorouracil, and this is a cytosine analog, and it interferes with RNA synthesis. So microbes are unable to, they lose the ability to produce the proper RNAs. So that means that the cells will stop growing. Unfortunately, fungi and other microbes are able to rapidly develop resistance to this drug. So it's not usually prescribed alone, but rather it gets prescribed as part of an antifungal regimen that includes other drugs. Let's do a quick check to see if you're with me. Here's one question. Which of the following inhibits ergosterol synthesis? Would that be griseofulvin, azoles, polyenes, flucytosine, or tolnaftate? Pause the video and press play when you're ready for the answer. Azoles, that's correct. Here's another. Which of the following is not a common target for antifungal drugs? Is that ribosomes, ergosterol, chitin or beta-glucans, or nucleic acids? What do you think? Ribosomes, that's right. Remember, fungi, like humans, have ADS ribosomes, so any drugs that we might use that would target a fungal ribosome would likely target our own as well. But what about viruses? Viruses pose a challenge for chemotherapeutic treatments because targets like the plasma membrane, cell walls, and ribosomes aren't present. Remember, although viruses appear to be living organisms when they've infected a cell, they're really non-living things. This table is here to remind you of some of the similarities and differences between viruses and living organisms. Although there are many antiviral drugs, most are designed to treat infections caused by HIV, herpes viruses, hepatitis viruses B and C, varicella zoster, remember that's the one that causes shingles, or influenza A and B. There are two main strategies for these antiviral treatments. The first strategy is to disrupt some step of the virus life cycle, preventing the virus from reproducing. The second strategy is to disrupt nucleic acid synthesis, which again is going to stop a virus from reproducing. Let's take a look at some of these drugs. Your first antiviral drug is acyclovir. Maybe you've probably heard of this drug before. In a normal mammalian cell, a nucleoside, so remember a nucleoside is basically a sugar plus a nitrogen-containing base. It gets converted into a nucleotide through the addition of a phosphate by an enzyme called thymidine kinase. This nucleotide can then be incorporated into a growing DNA strand by DNA polymerase. So in this example, we have a nucleoside, it's a guanine nucleoside. The phosphate gets added, making a guanine nucleotide, and then that guanine nucleotide gets incorporated into DNA opposite of cytosine. And everything's fine, it's hunky-dory, everything is normal. Now, in cells that are infected with viruses like herpes virus or Epstein-Barr virus or varicella zoster, in addition to normal cellular thymidine kinase and DNA polymerase, the virus will actually make the cell produce a viral thymidine kinase and a viral DNA polymerase. Now, acyclovir has been designed so that the viral thymidine kinase will bind it and bind a phosphate and convert that acyclovir into a fake nucleotide. Then the viral DNA polymerase will bind the fake nucleotide and try to insert it into a piece of viral DNA. Now, this will stop DNA synthesis by the virus of the virus. You might be wondering, well, why doesn't the DNA, the cellular DNA polymerase and the cellular thymidine kinase work with acyclovir? Well, the answer is they can't. Structurally, acyclovir has been designed for the viral proteins. Acyclovir isn't the only nucleotide analog out there. Tenofovir is another nucleotide analog, but this one is for HIV. Sometimes this molecule is called Truvada as well. And if you compare the drug here on the left to an actual nucleotide on the right, you'll see there's some similarities. We have an adenine base here and here. We have phosphate group here and here. We have a sugar group for our normal base, but there's no sugar group for the nucleotide analog. So again, Truvada is going to do the same thing. It's going to stop DNA synthesis for HIV virus. Now, just a couple of things about Truvada. Truvada is taken to interrupt the HIV life cycle, preventing viral replication. Keep in mind that this doesn't completely eliminate the virus from the body. However, it stops the virus from reproducing. If someone were on tenofovir and then they stopped taking it, or if the virus developed a mutation to tenofovir, then it would start growing again. Tenofovir is also used as a PrEP medication. So PrEP stands for pre-exposure prophylaxis, which means that the drug is meant to protect against HIV infection before someone comes in contact with HIV. All right, so that's Truvada, aka tenofovir. So remember, a few slides ago, I said that antivirals have two basic strategies. They either interrupt DNA synthesis, like acyclovir and Truvada, or they interrupt one step of the virus life cycle. So let's look at HIV as a model for the second type of antiviral. Remember, HIV has this unusual life cycle. It's an RNA virus made up of two strands of plus strand RNA. It's enclosed in a capsid and it's enveloped. There are spike proteins that are on the surface of the virus that bind a receptor called CD4 that's on the surface of your CD4 positive T lymphocyte cells. Once the spike protein binds CD4, it brings the virus close enough to bind a second co-receptor, an entry receptor called CCR5. When both receptors are engaged, the cell is stimulated to fuse the envelope of the virus to the plasma membrane of the cell. This begins the infectious process. Once the virus is inside the cell, it's going to uncoat, releasing viral RNA plus 3 enzymes into the cytoplasm of the cell. The first enzyme is called reverse transcriptase, and the job of reverse transcriptase is to bind the RNA from the virus and reverse transcribe that to make a viral DNA, which you see here. The second enzyme that's released is called integrase. So integrase takes the viral DNA, locates it to the nucleus, cuts the backbone of the cell, DNA, okay, so the backbone of the cellular DNA, and then it inserts the viral DNA right in the middle. So we get what's called a provirus. And the cell doesn't know the difference between the HIV provirus and any other cellular DNA, so it's going to transcribe the viral genes just like it would its own regular genes. So we'll get new copies of the viral genome. Some of these viral RNAs will be translated to make viral proteins. Some of those proteins will insert themselves into the plasma membrane of the infected cell. Some of them will go on to become a capsid. After maturation, where the virus has assembled at the plasma membrane, the virus will eventually bud away. So your anti-HIV drugs are going to target one or more of these steps. HIV replication can be blocked at a number of steps during its life cycle. For example, there are monoclonal antibody drugs and CCR5 antagonists that block HIV binding to CCR5 so that HIV cannot stimulate its co-receptor and gain entry to host cells. You also have fusion inhibitors that bind the spike protein of the HIV virus preventing it from undergoing structural changes that bring the viral envelope close enough to the host cell membrane to allow fusion. There are the anti-reverse transcriptase nucleoside analog style drugs like tenofovir that basically gum up the ability of reverse transcriptase to make a DNA copy of the viral RNA genome. Drugs like Stribild and Icentris bind integrase and prevent it from transferring DNA copies of HIV genome into the host chromosome. The final antiviral treatment that we'll look at are the interferons. Interferons are communication proteins called cytokines, and they're produced by the human body to prevent the spread of viruses from infected cells to adjacent cells. When a virus enters a host cell and it uncoats and starts reproducing, genes for interferon alpha and interferon beta will be synthesized and the proteins will be made. These proteins then get secreted from the infected cell, and they bind receptors in the plasma membrane or in the nucleus of adjacent cells. This binding triggers the production of antiviral proteins or AVPs in the adjacent cells so that if a virus tries to infect, the antiviral proteins are there ready and waiting to destroy the nucleic acids, for example, for the virus before it has a chance to actually infect the cell. This helps to heighten a cell's response to infection and provide extra defense. So again, your interferons prevent the spread of viruses from infected cells to uninfected cells nearby. And this is done through the activity of interferons alpha and beta. Now, interferon alpha has been extracted from human blood and purified, and you can purchase it. It's a drug called multiferon, and it's used to treat viral hepatitis patients. Basically, it's a preparation of multiple types of interferon alpha, and it can stimulate immune system cells like B cells and natural killer cells so that they will target the hepatitis viruses. Imiquimod is another drug. It promotes the production of interferons. And then this little note at the bottom is that interferons alpha and beta aren't the only interferons. There's another interferon called interferon gamma, and it causes neutrophils and macrophages to kill bacteria. So during a bacterial infection, this gets secreted to stimulate the death of bacteria. Here are a few more questions before we wrap up this video. Which of the following is not a virus that may be treated with antiviral drugs? Is that hepatitis B, RSV, herpes virus, HIV, or influenza? RSV. That's right. Remember, your antiviral drugs have been developed to combat hepatitis B and C, herpes viruses, varicella zoster, HIV, and influenza. Which of the following is not a target for anti-HIV drugs? Is that neuraminidase, integrase, CCR5, protease, or reverse transcriptase? Of course it's neuraminidase. We know that that's part of one of the spike proteins on influenza. It's not even part of HIV. Which of the following produces fake guanine nucleotides when acted upon by viral thymidine kinase? Is that rifampin, tetracycline, interferon, acyclovir, or neomycin? I knew you'd get that. Acyclovir. Great job. So in this video, we talked about a lot of antifungals. We talked about the polyenes, the azoles, the echinocandins. We talked about nucleic acid inhibitors as well. Remember, for your exam, you need to know what their targets are. For example, which of these is an ergosterol synthesis inhibitor? Which one binds ergosterol and causes the formation of cylindrical pores in the plasma membrane of cells? Which one is the penicillin of the fungal cell wall? And which one is a prodrug that becomes active in fungal cells and inhibits the synthesis of fungal DNA? I also described the two types of antiviral drugs. Remember, you have antiviral drugs that are nucleoside or nucleotide analogs. They mimic real DNA and RNA in some cases, nucleotides, but they stop the synthesis of nucleic acids. We also talked about drugs that inhibit different steps of the viral life cycle. For each type of drug, I gave you some examples that you'll want to remember for your exam. I hope this video was helpful to you. In the next video, we'll talk about resistance. I look forward to seeing you there.